So I would definitely word associate. A theory is broad, a law is generalized, and a hypothesis is an if-then statement. It's also very important for a hypothesis to be falsifiable, which means that it can't be tested. Testable is another way of saying that, but falsifiable is better in a way because it's not only saying that it's testable, it's saying that there's a way to prove if it's wrong. Like her question about flying potatoes in space. There's not really a way to definitively say there's not, because you can't search every inch of space. So that is testable, but it's not really falsifiable. Okay, so experiments. You guys just got done with research lesson 2. So I'm sure you are a little bit more familiar with an independent and a dependent variable. An independent variable is what you are changing. So I think independent, I can change it. And a dependent variable is what is being measured. So say you have a graph. Which of your axes is going to be the dependent variable? You guys can unmute yourself because I am going to be asking a lot of questions, so this is just not a lecture. So I'm sorry, but you're going to have to talk. Which axes? So sorry. Say that one more time. Which axis has the independent variable on it?
Can you hear me?
Okay. Yeah. Yeah, I can hear you.
The x-axis?
The x-axis. Beautiful. All right, so the x-axis has the independent variable. So that means that the dependent variable has to be the y-axis. And that's on a graph, and you guys just made 3 graphs, so you should know them backwards and forwards. The one thing on this slide that's kind of new is a controlled experiment, and that's an experiment in which one group does not receive the experimental treatment. So, I don't know if I have any Grey's Anatomy fans, but you all know that in a medical trial, some participants get the placebo and some participants get the drug. So the placebo is like the controlled. So they're part of the controlled experiment because you don't change anything. That gives you a baseline. An example that she used in class was if you wanted to see if fertilizer makes a plant grow taller. Okay, if I have a plant with fertilizer, how do I know if it makes it grow taller unless I have the same plant with the same soil and the same water and the same conditions, but with no fertilizer? Okay? Okay, so reasoning. You have inductive versus deductive. So these, in my opinion, are kind of confusing. I always got them confused when I was in y'all's shoes. I remember is deductive, detective. So what I do, I know she used the funnel example on if it's a funnel like this, it's deductive, or if it's an upside down funnel, it's inductive. I always like to try to give you guys something different to remember. So I remember deductive, detective. So if she will give you an example, say on an exam. And what I think when I read that example is, okay, could a detective take these pieces of evidence given and make this conclusion? If he can, it's inductive. If he can, it's deductive. If he can't, then it's inductive. That works a little bit better for me. But you guys, if the funnel examples work for you, if you're taking a lot of evidence, so you have your regular funnel, if you have a lot of evidence and make a generalized conclusion, so like one conclusion, then that is deductive. If you have an upside down funnel where you take one or two observations and make a generalization, say about like every, if I would say, I'm from West Virginia and my favorite color is yellow, then that means every single person from West Virginia's favorite color is yellow. That would be an inductive assumption. Does that make sense to everybody?
Will you explain how you said you determined them apart?
So I always think, okay, could a detective, so detective, deductive, could a detective find this evidence and come up to this conclusion? So say your example is you found a bloody knife in George's backpack, and come to find out his sister had been murdered. Can a detective take that information and say, well, George probably did it? Yes or no?
Yes.
Yes. Okay.
Yes.
So since they can, that is deductive. Now, I'll use my other example. Say I'm from West Virginia and my favorite color is yellow. So every single person from West Virginia's favorite color is yellow. Can a detective take that information, my favorite color is yellow and I'm from West Virginia, and come up with the conclusion that everybody from West Virginia's favorite color is yellow?
No.
Exactly. So when
no.
That's how I do it myself. I always think, can a detective do this? Does this make sense for a detective to do? Would I see this on like a cop show? And that makes it easier for me. Of course, the funnel, if you guys like the funnel idea, please use that. I just always try to give you like a second option. Okay, does that make sense? All right, moving on. Okay, vocab. You guys, I cannot stress enough how much this vocab is so important to learn. Very important to just kind of get a general feel for. So hydrophobic means sphere of water. So if a molecule is hydrophobic, is it polar or non-polar?
What'd you ask again?
Okay, so if a molecule is hydrophobic, would it be polar or non-polar?
I think it'd be polar. It's polar, right?
Okay.
No, because non-polar means it's like it's oxygen, right?
Yeah, exactly.
So I had that backwards.
But that's okay. Okay, so if something is hydrophobic, then it is non-polar.
So hydrophobic is non. Yes.
Yeah. Yep. Okay, now on the flip side, hydrophyllic means water loving. So is a hydrophilic molecule polar or nonpolar?
Polar.
Polar, exactly. So if she gave you just, okay, so if she said this molecule is hydrophobic, is it polar or nonpolar? She will expect you to be able to connect that. Now,
like if you have a diagram or like a biomolecule, whatever, the image of it, and she says, can you determine the polar or non-polar group? Like, how do you do that? Do you just like see if there's like OH in there, and if there's a lot of them, then you know it's polar, but if there's not, then you know it's non-polar? Like, I'm confused about that.
Exactly. Yeah, you're exactly right. So if she's saying point out the polar group, like in a phospholipid, that would be the phosphate group because that has a lot of oxygen. If it is a carbohydrate, then it's the carbon chain that has a lot of OHs. It's being able to pick out the polarity of a certain molecule is just being able to see, okay, where's the most oxygens at? Does that make more sense?
Yeah, thank you.
No problem. Okay, so of our known biomolecules, which one is characteristically known for being hydrophilic?
Carbohydrates, because they dissolve everything.
Exactly. All right. Now, on the other hand, which ones are characteristically known for being hydrophobic?
Lipids.
Beautiful. Okay, so dehydration synthesis. Sorry, did somebody say something? If you did, that's totally fine. I kind of cut you off. Oh, somebody said they can't hear me. Can everybody else hear me? Or no?
Yes.
Yes? Okay. All right, just making sure. Okay, gotcha. All right, thank y'all. Okay, so does the difference between hydrophobic, hydrophilic make sense?
Yeah.
Awesome. All right, so now on to hydrolysis and dehydration synthesis. Okay, so if I had one big molecule and I wanted to make two little molecules, so I had one big disaccharide and I want to make two monosaccharides. Let me click off of that. Okay. One disaccharide and I wanted to make two monosaccharides. What process would I have to undergo?
Hydrolysis.
Beautiful. All right, now say I changed my mind and I don't like these two monosaccharides and I want to go back to a big disaccharide. What process do I use?
Well, so if you did square root of 2 plus x times the square root of 2 plus x.
Dehydration synthesis.
Exactly. So I know she likes to talk about Jane and Carl, and I did not put a picture of this on here for a reason, because I think if you guys get too focused in on the actual chemistry of it, then it freaks you out and you're like, oh my gosh, I could never do this. Because I can totally understand how it could be overwhelming. So let's just think of this in conceptual terms, okay? Water is the key. Water can make or break a bond, okay? That's it. It's just water. Now, who knows the chemical composition of water? is H2O, right?
Yeah.
Exactly. Okay. Exactly. So, theoretically, it makes sense that when you break a bond, okay, so two molecules are always held together by oxygen. So when you break a bond, you add in H2O, and then where the molecules were once held, they now just have a hydroxyl group. So this one has a hydroxyl group, and this one has a hydroxyl group. Does that make sense conceptually? Because I understand if you guys see like molecules, then it starts to get a little more scary. But follow the hydroxyl groups. And if you get confused, think, how can I make water? How can I either put water in and what's it going to do, or how can I take water out? Does that make sense? Somebody please tell me. I will not get offended.
You're saying,
Mm-hmm.
but say it again. Just I'm like trying to make it click.
Okay, so hydrolysis. You have one big disaccharide, okay? Right? Okay, and you add water. You know what? I had a pretty good example of this the other day. You guys can see me, right?
Yeah.
And this little yellow scrunchie here is an oxygen, okay? So now they're holding hands by this little yellow scrunchie. That's an oxygen, okay? Now, water is added in and it's broken. This oxygen does not go away. It's just added a hydrogen. But this other little band comes on as another oxygen for this one. So now you have a hydroxyl group on each of your separate monomers. Does that make sense?
Yeah, that makes sense. Thank you.
No. Reversely, on dehydration synthesis, here's your oxygens. Now, remember, each oxygen has a hydrogen on it. I'm just running out of ways to show it. So, each yellow band represents a hydroxyl group. Now, dehydration synthesis, you want to take a water out. So, I'm just going to throw this one, and I'm going to get hooked again like this. So, you throw the composition of water out. So H2O. You throw it out, and now this oxygen that was on this hand is now connecting these two hands. Does that make more sense to you guys? Because I completely understand how the organic chemistry of it is totally freaking you out. So just think of it like hands and hair ties. Is that more clear?
Yes.
Awesome. Okay. All right, so now a monomer is just one. Mono means one. A polymer is many. Let me put my hair back up. Okay, so a polymer is just many, and a monomer is one. So the monomer is most of the time the simplest unit. So like a monosaccharide, that would be the simplest unit of a carbohydrate. Okay, does all the concepts on this slide make sense? Okay. Now, functional groups.
Sorry. Do polar things like other polar things, or do polar things like non-polar things?
You know, that is a good question. I want to say that polar things like non-polar things, like a magnet. but I'm not 1,000% sure. Let me get back to you, and I will include that in my next email to you guys, okay?
Okay, thank you.
Okay. I'm going to write it down or I would forget. Now, we do know that nonpolar things attract because you see this in like the phospholipids where the nonpolar tails are tucked in, right? So nonpolar things attract. But I'm not sure if polar and nonpolar attract or repel. I'm going to get back to you guys on that, okay? Okay, so functional groups. You guys are going to have to know what these look like. Not just for this class. I am a junior. I'm in a 400 level class, and I'm still learning about these functional groups. I'm sorry. I know they're a pain to learn, but if you learn them now, it's going to make your life so much easier. So, hydroxyl. Oh, I just told you the name of it. Okay, number 1 is a hydroxyl. It is literally just an OH. So it has oxygen and a hydrogen. Hydro, hydrogen, oxyl, oxygen. I think that one's pretty self-explanatory. Now, who can tell me what number 2 is?
Is the carbonyl?
Yes, awesome, awesome. Okay, so number 2 is a carbonyl. And a carbonyl is just a C with a double bonded oxygen. That is the key to this. It has other stuff on it, but the key is that the main distinguishing factor is that it's a C with a double bonded oxygen, and nothing else is like important. Okay? Now, what's number 3?
Is that a carboxyl?
That is a carboxyl. Now, you see 2 important things on the carboxyl group. You see the C to a double bonded O, but you also see the C bonded to a hydroxyl. So, how I distinguish between the two, I've actually heard it done 2 ways. The students last year told me about this way. So, if you guys can remember that this group is called a hydroxyl, a carboxyl has a hydroxyl. So this right here is a hydroxyl. So hydroxyl lives on a carboxyl. Okay? If that makes no sense to you guys, how I remember it, so you have carbonyl and carboxyl. The one that sounds like oxygen has the extra O. So this one has 2 oxygens. This one has 1 oxygen. So the one that has the most oxygens sounds more like oxygen, if that makes any sense. So because this has 1 more than this, it sounds like oxyl. It sounds more distinguishedly to be like oxygen. Does that make sense? So you can remember that a carboxyl has a hydroxyl, or you can think that a carboxyl has 2 oxygens. Okay? But no matter how you remember it, you need to know these. Okay? All right. Now we're going to start to get into the meat and potatoes. So first, let's talk about the carbohydrate. So carbs, their main functions are fuel and building blocks. We know this. We eat carbs in all the good food, pasta, bread, stuff like that. Let me blow up this image real quick. So the distinguishing factor, if you can see, it has a group right here on top. This is a carbonyl group, okay? Because you see the C So double bonded O, nothing else that looks exciting. You have a hydrogen and you have another carbon. Those really aren't that exciting. So the main thing is you have that C to the double bonded O. The next distinguishing factor that is of a carbohydrate is you have a lot of hydroxyl groups. OH, 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 OH. You have an OH, an OH, an OH. If you have a lot of hydroxyl groups compared to carbons, so you have 1, 2, 3, 4, 5, 6 carbons, and you have 1, 2, 3, 4, 5, 6 oxygens, then it's polar. Now, I'm not saying that has to be equal or has to be more. It just has to be a lot. And you can distinguishably look at this and say, like, oh, yeah, that has a lot of oxygen on it. That's polar. And if you can just look at that and say, oh, yeah, that's polar, then you'll be able to say, hey, that's a carbohydrate because it's polar. Because our other two biomolecules, the proteins and the nucleic acids, look a lot different. So the two that are really hard to distinguish between each other are carbs and lipids. So, do carbohydrates mix with water? Why or why not? Somebody who hasn't talked, which I don't know who's talked and who hasn't, but I won't bite, I swear.
Yes, they do.
Okay, why?
Because they dissolve water, and like if you have a lipid, they don't play well with water.
Yes, it does.
Mhm.
You have two.
That's really the only thing I know.
Okay, think about the molecular composition of it. So is it polar or non-polar? A carb.
Polar.
Polar, exactly. Okay, so since it's polar, that means that it is going to mix with water. Because if it's polar and it mixes with water, what's the fancy term for that again?
Hydrophyllic.
Awesome job, guys. Awesome job. Thank you for being talkative. I'm having a lot of fun. I don't feel like a lecturer anymore. Okay, so moving on to our second. We have lipids. So lipids have a lot of hydrocarbons. And what hydrocarbons are is a carbon with hydrogen on it. Hydrocarbon. It has a carboxyl group. So it has a C double bonded to an O with a hydroxyl on the end. But then it doesn't, so once you get away from that carboxyl group, it doesn't really have that much oxygen on it. Now, lipids do not have a lot of oxygen. They do have some oxygen. So if you get a picture of something on a test, even though it has some oxygen, does not make it automatically a carbohydrate, because lipids will always have at least 2 oxygens because of the carboxyl group. I've had so many people miss the test question because they look at it and they're like, oh, well, there's oxygen on it, so it's polar, so it's a carb. Great that they're connecting that, but even lipids have oxygen. It's not a lot. Okay? Okay, next questions. Do lipids mix with water? Why or why not?
Did you write that down? The lipids?
No.
You have Y.
Okay. Why not?
And discontinuity.
Because they're non-polar, which means they're hydrophobic, and hydrophobic means water-fearing.
Exactly. Beautiful. Okay, now types of lipids. We really have 3 types of lipids that we care about. The first is fats, and this is just your regular hydrocarbon chain. So this is exactly what I showed you. You have the carboxyl, and then you have your hydrocarbons. Now, there are 2 different types. There are saturated and unsaturated. The top is saturated, and with all these questions, I will show you guys there's an answer slide on the back. I put these on here so you guys can like test yourself if you want to. So here, the top one is saturated. And you can look at this and say, well, okay, of course it's saturated. There's no bends. Or right here, this is unsaturated. It's very clearly unsaturated because you have this bend. It looks almost like a broken finger or like a fish hook. It's bent. And that's because it has two double bonded carbons. Okay? All right. Now, phospholipids. She always says in class that proteins are our favorite biomolecule. But I will tell you that on exam 1, her favorite biomolecule is phospholipids. I haven't looked at the exam yet, but I've been doing this for a while, and I'm telling you guys, she loves phospholipids. So, phospholipid has a polar head and a non-polar tail. It contains fatty acids, glycerols, and a phosphate group. So, the phosphate group is what makes the head polar. The fatty acid tails, it has two tails, one saturated, one unsaturated tail, which is not really important, but it does. Those are non-polar. Okay, so what do phospholipids make up? They make up something really important.
Don't they make up the cell membrane?
Yes, ma'am, they do. And I think we're going to start our cell chapter on Wednesday, so we will learn a lot more on this on Wednesday. But phospholipids do form the cell membrane. Okay, so steroids are lipids too. And a lot of people will just see a ring structure on an exam or like somewhere else in life and be like, oh, well, that's a carb. Steroids are lipids and are rings also, but they still don't have that much oxygen. That's the whole thing. So let me see if I can blow this up. All right, so if you look on this picture, you have one oxygen, two oxygens, and all these carbons. See? So you still only have two oxygens compared to all of these carbons. So you still are not seeing a lot of oxygens. So steroids are always 4 fused carbon rings. And they're important in the body because they don't dissolve in blood. So they act as hormones. So say your brain was trying to send a message to somewhere else in the body. Wouldn't it be useful if that message didn't dissolve in blood? Like if it didn't dissolve in your blood and like halfway through your body it just dissolved and message never gets sent. Message failed. No Wi-Fi connection, can't help you. Which if it's trying to tell your heart to beat or trying to tell your whatever, then that's an important message that shouldn't get lost halfway through. Okay? Any questions on lipids or carbs? Okay, now we're going to move on to proteins. So proteins are the most versatile. They are mainly enzymes. They are used in the cell for fuel sometimes. They're very, very useful. Now, proteins have 3 major groups: an amino group, a carboxyl group, and an R group. Now, I know she lectured on the R group, but all you really need to know about the R group is that anything can be there. A polar group can be there, a non-polar group can be there, and an amino group and a carboxyl group kind of cancel each other out. So the R group does determine polarity. Okay? Okay, guys. So I don't know if you guys can see, but I have like a green box around this group. What group is in the green box? So this nitrogen with the 2 hydrogens. What functional group is this?
Difficult sometimes.
Is that amino acids?
It's an amino group. So this whole thing is an amino acid. The amino acid is the simplest form of a protein. It's kind of like a monomer of a protein. But this is an amino group. Good job. Okay, so this one we've already seen. What's this one?
Is that the carboxyl group?
Sure is. All right, and then there's a hydrogen on here, which we don't really care about, but it's there. Okay. Now, we're going to move on to the levels of organization of a protein. Very, very, very important. You need to know which levels have which bonds. So, primary have peptide bonds. So, P and P. It goes together. Primary peptide. Okay? And they're flat. So they're literally just a line of amino acids. They're just lined up. That's all they are. They're just flat and simple. Okay? Now, secondary is when you start to get the shapes. So you get the alpha helix and the beta pleated sheets. So say here's your primary level. Here's your secondary level. That's supposed to be a pleat, but it looks like an L. But you guys get the drift. It's not perfectly straight anymore. You have like kinks. Okay? And they're held together by hydrogen bonds. Okay. I have tried and tried and tried to find a mnemonic for secondary and hydrogen, and I cannot. But I have a mnemonic for all the other ones. So, worst comes to worst, process of elimination. I have tried to find a mnemonic for secondary and hydrogen bonds. So, if you guys can come up with one, please share. Okay. Tertiary. This is the first level that is 3D and a functional protein. So a functional protein, tertiary level, and it's 3D. So that's when like your hands are balled up. Or, okay, never mind. All right, tertiary is like one fist. It's 3D. It's one fist. Okay? Now they're held by side chain interactions, and that's all you need to know. You don't need to know if it's polar, non-polar, charged. No. You just need to know that they're held by side chain interactions. So my mnemonic for this is Terry is a side chick. Tertiary held by side chain interactions. Terry is a side chick. I know, but it works. Okay? So quaternary, I can never say this word, and I promise you I will say it different every single time. So literally just bear with me. And quaternary is multiple polypeptide chains balled up. It's also a functional protein, but this is like a complex protein. So a tertiary is just one fist. Quaternary is like if you put both of your hands together like this. That's an example of like a quaternary protein. And they are held together by ionic or hydrogen bonds. I should flip these because of my mnemonic. So they're held together by hydrogen or ionic bonds. And my mnemonic for this is, I wish I had a quarter for every high I heard. A quarter for every high HI. I know it's kind of not that great, but it's all I could come up with for this one. So if I had a quarter for every high I heard. All right? Does that make sense to you guys? Any questions? Okay. Now, I know we barely grazed the surface of nucleic acids, but I'm going to go ahead and talk about them. And if you don't understand, that's totally fine. I just want to kind of graze a little bit. Okay? All right, so nucleic acids, they all contain a 5-carbon sugar, a nitrogenous base, and a phosphate group. Now, here's a picture of one, and as you can see, it looks very complex, and it looks nothing like a protein, a carb, or a lipid. So they're all very distinct. So this whole thing is a nucleic acid. Okay? All right. So there's 2 types of nucleic acids. You have DNA, which is double-stranded. So DNA is double-stranded. And that has long-term storage. On the other hand of this, does somebody have a question?
3 on the left.
Okay, so on the other hand of this, the RNA is single-stranded, and it aids in gene synthesis, regulation, and expression. So how I like to think of this is that the DNA is the person that just sets and commands people. So I like to think of DNA as the big boss. Big because it's double-stranded, and it kind of just hangs out in the nucleus and tells things what to do. An RNA is like the working man. It's a little thinner because he's a working man. He's single-stranded, and he goes out and he gets stuff done. He does stuff. He doesn't just stay at home and give orders. He actually goes through with the orders. Okay? Does that make sense? All right, so these slides are on cell theory, which we have not started yet, but I'm still going to send this PowerPoint out with this information because I'm sure we'll get to this by Wednesday. So if you have any questions about cell theory stuff, there is some like check yourself questions on here. The very last slide is the answer slide. So if you have any questions, just let me know. Okay? All right, I'm going to stop sharing. Okay, so do you guys have any questions? Is there anything that's really not clicking with you? Something you want me to go back over?
And or any other discontinuity using analytical techniques.
I have a few questions, but they're kind of from the lab. But it's like questions that I think you could probably answer.
Okay.
But let me find it. So on the lab it said, from the biomolecules listed below, choose one that is most likely to be polar or is most nonpolar.
Okay.
So then the choices that was amino acid, steroid, monosaccharide, phospholipid, and nucleic acid. So like out of all of those, I chose that a steroid would be nonpolar because it's considered a lipid, and because it would have like less oxygens than a phospholipid would because phospholipids are like half and half. But then I didn't like I could pick which one was most nonpolar, but then she says for the wrong answers draw them out and circle the polar group, and like I'm confused how to determine the polar group in a nucleic acid or like an amino acid. Like I don't know where the polar group is in the drawing.
Gotcha. Okay, so a nucleic acid. Hold on, let me share my screen again. Okay. So here's a nucleic acid. So 5-carbon sugar, we haven't really talked about that. Nitrogenous base, haven't really talked about that. Phosphate group we have talked about, right?
Yes.
Okay, where have we talked about a phosphate group before?
In the phospholipid.
Okay, and
Like,
where
which is
did the phosphate group live in a
in
phosphate?
the head of it, the polar head.
The polar head.
Yeah.
See where I'm going with this? Okay. Now, what was your next one?
But she says to like circle the polar group, like in that drawing. So what would I circle? Just like all of the options? Okay.
Okay. So a group is a functional group. So the groups of nucleic acids are the 5-carbon sugar, the nitrogenous base, and the phosphate group.
Okay.
Okay.
And
so
then
right here.
The amino
that
acid.
one. Then it would be the same for a phospholipid. I would just circle the same group as I circled in nucleic acids.
Yep.
Right here.
Okay, and then the other one
Right
was
for those two.
monosaccharide. Like for that one, I would just circle all of the OHs, correct?
Then we looked over on this side.
Yeah.
Make these two.
Yep.
Because like I understood the pictures, I just wasn't sure since like they were half polar and half non-polar. I didn't know how to go about circling those or whatever.
My brain is just there.
Yeah.
And then I had one more question. Let me find it. How do you identify the type of bonds that like holds a biomolecule together? Like
all that we're left with 6 minus 3.
Like if it's an ester bond or
Plus 1.
I'm still confused on that.
Right here.
Okay, so a protein will always be a peptide. Well, not always, but the primary level protein peptide, that type thing. And then we just went through a big list about the others, the other levels. A carbohydrate most of the time is just going to be a covalent bond.
Okay.
And a lipid, specifically a trisaccharide, a triglyceride, not a trisaccharide, my bad, is mostly going to have ester bonds.
Okay, so like,
got canceled. You canceled that out.
For a peptide bond, like an amino acid would be more likely to have a peptide bond.
Would it? What do you think?
-3.
Well, I think an amino acid and a protein, like the coordinary structure of a peptide bond.
Look for if this part down here equal to 0.
But I
At the
didn't.
bottom equal to 0.
But it says I have to choose between both of those.
Then you know.
Of which is most likely to have one, and then that just confused me because I was like, they both have one.
They both have one, but you're mainly associating primary with peptide.
Okay.
Yeah, so I'm sorry. If you guys don't have any more questions, you are free to go. I just took a picture of your attendance. If you guys know of anybody that was at the session but did not stay until I literally just snapped the picture 2 seconds ago, just have them email me with the color of my shirt and I'll count their attendance, okay? All right, and if you guys have questions, feel free to stay. If not, I will see you guys on Wednesday.
You have to separate them like this.
I think that pretty much covered it. So, like, phospholipids, they're like, would they have an ester bond too since they're a lipid?
Negative 1.
That I'm not sure of. I would think so because of the fatty acids.
Okay.
But I will tell you that, like, on an exam, she's probably not going to ask about phospholipid bonds.
Okay.
Plus
But
Because I
I'm going to write that question down to ask, and I'll put it in my email when I send you guys out this PowerPoint.
Yeah, because on this lab, since I'm in Group B, like we started on week 4 lab, and like we haven't started on the week 4 lecture
plus
for Miss Arnett, so I was confused at what some of the stuff meant because we hadn't learned about it yet.
Yeah, no, I totally understand that.
up here.
So I was getting most of my answers from her PowerPoint
Out of all this.
for lab, but I still didn't. I didn't know what my answers meant because we hadn't went over it. So I was like, that was when I was emailing you the other night because I was just so had no idea what I was doing.
You're absolutely fine. That's totally okay.
So I think that
You take out an X.
pretty much covers it.
X cubed minus 2.
All right. Well, you know, if you need anything, shoot me an email, make a tutoring appointment, holler at me across campus. You'll be able to down somehow, okay?
Thank you. You're really
No
good at
problem.
helping and explaining, and a lot better than she's like way over my head. Yeah.
I totally understand that. All right. Well, you have a good day, okay?
You too. Thank you.